In this tutorial we will discuss pulse laser and its principle. Let's discuss with introduction. The time-resolved spectroscopy or pulse laser technique is a latest technique to study the rate of or dynamics of chemical reactions initiated by the absorption of light. Chemical reactions that result from the absorption of light are called photochemical reactions. Some examples are photodissociation, photoisomerization, and photodimerization, etc. The time required for completion of such photochemical reactions are too fast in pico or femto range, that is in the range 10 to the power minus 12 to 10 to the power minus 15 second. To study the rate of such fast reactions, we use femtosecond or picosecond pulses. In time-resolved spectroscopy, laser pulses are used to obtain the absorption, emission, or Raman spectra of reactants, products and even transition states of reactions. Principle of pulse laser. When sample A is treated with fast pulses, then it gets excited to an electronic state A*, that can emit a photon, as fluorescence or phosphorescence, or react with another species B to yield a product C. The atom A plus H nu goes to excited state A*, by absorption of the photon H nu. The excited state atom A*, goes to ground state atom A by emission. Or, the excited state atom A*, combine with another species B to form product C by reaction. It is clearly mentioned here. The quantum yield of photochemical reaction is phi is equal to number of molecules that undergo reactions by absorption of photon by number of photons absorbed. The value of quantum yield varies over a wide range. For example, phi is similar to 10 to the power minus 3 for the bleaching of certain dyes. Phi is similar to 1 for photodissociation of ozone. And, phi is similar to 10 to the power 6 for the reaction between hydrogen gas H2 and chlorine gas Cl2. In time-resolved studies of the photochemical reactions, the light source is a pulsed laser, which generates light pulses of short duration that is similar to 1 into 10 to the power minus 14 second or 10 femtosecond. The laser output is split into two parts by a partially reflecting mirror or beam splitter. The laser output is now divided into pump pulse and probe pulse. The direction of each pulse is determined by mirrors. The pump pulse is used to initiate the photochemical process in a sample, and the probe pulse is used to how the sample changes in response to the pump pulse. If the lengths of the two paths are same, both pulse arrive at the crossing point in the sample at the same time. In this case we say that there is no time delay between the two light pulses. If the path lengths differ, the two pulses will arrive at the sample at different times. In time-resolved spectroscopy, some property of sample, that means intensity, is measured as a function of the delay time between these two laser pulses. Let us consider the photodissociation of iodine cyanide ICN gas as iodine cyanide ICN gas plus H nu goes to iodine gas I and cyanide gas Cn. Like as mentioned here. For this reaction the pump wavelength is set as 306 nanometer and the probe wavelength is set at 388 nanometer. Finally, the iodine cyanide gas ICN is excited from ground state to the excited dissociative states by pump pulse at 306 nanometer. Upon excitation, iodine gas I and cyanide gas Cn are produced. When probe pulse at 388 nanometer is subjected to cyanide gas Cn, it gets excited to excited Cn* state. The excited Cn* then decays or gets relaxed, and subsequent fluorescence from excited Cn* gas is then recorded. The intensity of the fluorescence caused by the probe pulse is a measure of the concentration of cyanide Cn gas present in the sample. If the intensity is plotted as a function of the delay time between the arrival of probe pulse and the 306 nanometer pump pulse at the sample, we get the curve as shown in the figure. The solid line is a curve fit of the data to the function. Intensity I is equal to 1 minus e to the power minus t by tau, where t is equal to time delay of the pulse, and tau is equal to the pulse width. This much for today. Thanks for watching. Bye bye, see you.